Well, thank you so much for that introduction and the opportunity to speak. I feel really lucky to have the chance to share a little bit more about what my lab has been doing and to understand the effects of salt on freshwater communities. And so before I get started, I wanted to take a second to recognize that this is a complex issue, trying to balance the safety of the roads and also what road salts do to the environment. And so I wanted to start with an illustration. I'm in Madison, Wisconsin, and last winter we had an incredibly tough winter storm. And this past fall, I had the privilege of interacting with people at Madison Streets Division, and I learned a lot about what Madison citizens thought, their opinions about road salts, and it's everywhere, as you all probably are well aware. There are folks that have the opinion of we need to use more road salt. For example, this is a message to the mayor. Mayor, the streets are a mess. Use more salt. Safety is just as important as water. So we have those that want more salt. And then we also have those that want less salt. So, hello, Mayor. By using less salt, you are keeping literally hundreds upon hundreds of tons of salt out of Madison's lakes and groundwater. This will benefit countless wildlife, including us. And so, yeah, this is a complex issue. And the more I learned about it from the perspective of Madison Streets Division, I just felt so grateful for the people who have to navigate this day in and day out. And so, another reason for me to put up these sort of examples is my way of sort of helping me frame the point of this talk. And so, in this talk, I'll be focusing more on that second opinion. So, thinking about what road salts are doing to the environment, specifically freshwater ecosystems and the organisms within. And so, my goal for this talk are twofold. The first, I want to describe a little bit more about how we approach understanding how road salts affect the environment and wildlife. And then two, I want to give some information about some of the discoveries that my lab have found in the past few years about the effects of road salt. And so with that, I wanted to start with this traditional toxicological approach. When we're trying to understand what road salts do to wildlife, one way to do this is to assess the direct toxicity of road salts on model organisms. So in this image here, you have two model organisms. You have a fish and then you have a water flea. The idea is that if we understand what road salts do to the fish or what road salts to this model invertebrate, we can then extrapolate what road salts are going to do to the rest of the community. And so there are many different benefits to using this type of approach, right? They're easy to run, they're standardized, and it's easy to replicate across time and across different species. And so while there are many benefits, the idea is that we use these short-term lab tests, these traditional toxicological approaches to predict toxicity in natural communities. So we use models to extrapolate to all the other organisms in the system. Now, despite the benefits, there are also many important limitations. And probably the most central limitation that we think a lot about is that this approach ignores a lot of the complexity that we actually see in nature. And so a central theme in my lab and what a lot of my lab group thinks about is how do we integrate in that ecological complexity into our understanding of road salt? So this is the roadmap for today. What I'll do is I'll start as simple as we can, asking what are the effects of salt on the species level? So across similar species, if we use a model organism, if we understand what salt does to one species, does it help us understand what salt does to the rest, to other very similar species? From here, I'm going to start layering on more and more complexity. So it's not about just different species, but even within a species, these organisms are living in very different environments. Can we understand what salt does to one environment? Does that necessarily help us understand what happens in a completely different environment? So we'll talk about it at the population level. Next, we all recognize that these organisms aren't existing in a vacuum. They are interacting with a number of other organisms in their community. And so in this third section, I'll talk about how salt can influence these interactions. And then lastly, salt is not the only chemical that's in the environment. In different ecosystems, there are other non-living factors like water quality that might either exacerbate the negative effects of salt or even dampen the effects of salt. And so this is what you can expect in today's talk. I'll start simple and layer in that ecological complexity. Okay, so I'll jump right into that first question. I mentioned that traditionally what we tend to do is we choose a model, we understand what salt does to that model, and then use that to help us understand whether a system is going to be threatened. In this first section, we'll challenge that approach. We'll ask, are the effects of salt generalizable across similar wetland species? So across similar species, can we even make those predictions? To address this question, I'll talk about a traditional test called the LC50 test. This is a very standardized toxicological approach where we ask what is the concentration that is lethal to 50% of test organisms. And that's just an image of what it tends to look like. So it's really sterile, it's controlled, we remove all variation except for the road salt and the organism. And so how it typically works, you have organisms in a dish, and we expose these organisms to higher and higher concentrations of road salt. We might predict that at the highest concentration, this is going to cause the most mortality. At the low concentration, no mortality. And so we can take this and graph it. So when you look at the graph here on the x-axis, that's the increasing concentration of salt. On the y-axis, this is the response survival. So it looks something like this. At low concentrations, high survival. At high concentrations, low survival. The LC50 value is that concentration that causes 50% lethal effects. So in this example, 100 parts per billion, that is the LC50 value. And so the value of tests like this is that we can calculate these LC50 values for a number of different species and then compare the LC50 values to understand relatively which of the species are more susceptible to others. And so I'll say this again in the next few slides, but a high LC50 value, that means it takes a lot of road salt to cause 50% mortality. So those organisms are relatively tolerant. A low LC50 value means it takes very little to cause 50% mortality. So that means that organism is relatively susceptible to road salt. So thinking now about organisms, I am trained as a herpetologist, so I think a lot about amphibians. And there are many reasons to study the effects of road salt on amphibians. One of which is that there are over 8,000 species of amphibians worldwide, and a third of them are threatened. And actually, as of last year, 41% of amphibians are considered threatened. This makes them the top most threatened vertebrate of all across the world. And so understanding what pollutants like road salts do also has broad conservation implications. With that, even if you don't care about amphibians, amphibians are incredible models because they serve as a sentinel for what might happen to the other organisms in that system, like fish or other organisms that people might care about. The reason for this is that they have highly permeable skin. So they're incredibly susceptible to pollutants like road salts, which make them a great indicator species. So if we understand what happens to amphibians, this can help us understand what might happen to other organisms in that system. So using amphibians, again, the question is, are the effects of salt generalizable across very similar species? To do this, we studied the wood frog, the spring peeper, the leopard frog, and the American toad. And so what's going to pop up on the screen are LC50 values. Again, a higher LC50 value means they're more tolerant to salt. A lower LC50 value means they're more susceptible. And the point of all this is to ask, if we understand toxicity to salt to one species, does that help us understand toxicity of salt to other organisms? And as the data pop up, I hope that one of the things it conveys to you is that there is a ton of variation. Understanding toxicity to one of these tadpoles, one of these amphibians, doesn't necessarily help us to understand how toxic it might be to another one. And so wood frogs, for example, are highly susceptible. If we were to choose wood frogs as our model and look at regulations on what salt is toxic, we're going to completely overestimate the toxicity of salt. So we're going to say that it's going to take four to cause 50% mortality, whereas if you look at the toads, it takes eight, right? And so taking a step back, you might be wondering, what do these numbers mean? So for some context, 18 grams per liter, this is the concentration that we find in some of our samples with runoff. The highest concentrations in wetlands were about 4.6 grams per liter. And then most of the ponds that we sampled range anywhere from 0 to 1.1 grams per liter. So what does this mean? It means that at the most common concentrations, these concentrations are not large enough to cause mortality for most amphibians. However, in the worst case scenario at 4.6, the vulnerable species, the wood frogs and the spring peepers, may be susceptible to runoff from salt. And I'll come back to that and why that is just the first step to understanding negative effects of salt. So far, I've shown you some data. Salt can cause mortality for the most susceptible species, wood frog and spring peepers. But in the worst case scenario, so only in those situations where it was the highest amount of salt, in this study, it was unlikely to cause direct mortality road salt at environmentally relevant concentrations. And I hope you remember this conclusion because I'll come back to it and talk about why, yes, this is one perspective, but is way more complex. And it's not just about the concentrations that cause mortality. So we'll come back to this conclusion. The main take home I'd like to convey is that we use this system where we focus and rely on models to make predictions about what other organisms are going to face when given a contaminant. And I think what studies like this demonstrate is that considering species level variation is really important to understanding how organisms are going to respond to pollutants. Because if we choose a susceptible species, we're going to overestimate how toxic that chemical is. If we choose a tolerant species as our model, we're going to underestimate. And so care needs to be taken in order to be accurate about the effects of red salt. So that being said, one of the main limitations of this study is that when we're running toxicological assays, we tend to try to simplify and control as much as possible. And the reason for this is we want these tests to be repeatable and reliable. The negative thing about this is that the subjects of LC50 tend to have limited genetic variation. This is a problem because when we look out in nature, these organisms are living in very different environments that can cause selection that can increase genetic variation in the organisms that we care about. And so some organisms may live in a pond right next to a road where they're exposed to salt. Others might live in more relatively pristine environments where they've never experienced salt. Considering this variation may be important in understanding and choosing models to help us understand how toxic salt will be. And so in the second section of this talk, I'll highlight how generalizable is sodium chloride across populations. So in the first part, we're talking about across very different species. Now we're talking about the same species, just living in different areas. And so specifically, we'll talk about the wood frog and wood frogs living in 15 different locations across northwestern Pennsylvania. So what we did was we looked at wood frogs and we tested across these 15 populations just variation in how much they could tolerate road salt. On this figure here, on the x-axis, this is time. On the y-axis, this is percent survival. And so what I'd like to orient you all, anything to the left of the graph indicates that these tadpoles are highly susceptible. They're experiencing mortality from salt much faster. Anything to the right means that they are more tolerant. They are experiencing mortality when exposed to the same concentration much later. And so here are some of the data. I recognize that that's a lot of lines. And so what I'd like to do is draw your attention to those red arrows. Of organisms of the same species, the only difference is where they're found. From the most susceptible to the least susceptible population, there's a 61% difference in susceptibility. And so again, why does this matter? There is high variation in susceptibility to salt across populations. We need to take care in making conclusions based on single population studies. Again, this goes back to if we're choosing the wrong model, this can lead to really inaccurate predictions about how toxic road salts will be. Okay, so The first point is we need to consider variation. As we transition now to adding in more ecological complexity, one of the things to think about is so far, all of these tests have been these organisms in isolation. That's just not the reality. These organisms are interacting with a wide range of other organisms that can cause stress. So these tadpoles are interacting with predators, parasites, and even other competitors. And so in this section, I promised that I'd come back to the idea that these concentrations found in nature are not lethal. In this section, I'll talk about why that still matters in protecting wildlife. Specifically, the question that I'll talk about here is, can low levels of sodium chloride modify the way that organisms interact with other organisms in a negative way? Specifically, the question will be, at low concentrations, environmentally relevant concentrations of salt, does this make tadpoles more susceptible to diseases? So the pathogen and the parasite that we'll be talking about is the trematode. The trematode is a very common parasite found in freshwater ecosystems. It is a free-swimming parasite. It'll swim through the water column until it finds a suitable host. In this case, it's a tadpole, and then it's going to insist in the kidney tissue of the tadpole. This is a dose-dependent parasite, which means that the more parasites in it, the more negative effects it will have on the host. Specifically, we'll focus again on the wood frog. So the question is, does salt modify this interaction between the parasite and the host? To test this question, what we did was we exposed wood frog tadpoles to 4 different concentrations of salt. I wanted to point out that these 4 concentrations, these are concentrations that are environmentally relevant. These are the concentrations that are commonly found in ponds, right? They're low. They're not going to cause mortality to the wood frog. Then we challenged these tadpoles with 50 trematodes. This is that parasite that I talked about earlier, and we wanted to know, how does exposure to environmentally relevant concentrations of salt influence this tadpole's susceptibility to a very common parasite that it encounters in its environment. So on this figure, on the x-axis on the bottom there, we're talking about sodium chloride concentration. On the y-axis, we're talking about the number of parasites that got in. And so in every single sublethal concentration of salt, we find that exposure to salt increased tadpole susceptibility to trematodes. And so the take home that I'd like to convey here is that salt can modify host-parasite interactions. So in my field, there's a lot of emphasis in thinking about what is the concentration that is going to cause mortality. But what I think studies like this demonstrate is that it's not always about those lethal concentrations. These low concentrations that get in, they may not cause mortality, but they can still have negative effects by leading to negative consequences when it comes to dealing with other stressors like parasites in the system. And so in these studies, we tend to go from, we tend to use these incredibly highly controlled studies, which are useful, when in reality, the world looks a lot more like this. And so where we are now, I've talked to you about different models and why we need to consider variation across species and across populations. I've talked about how it's also important to think about those low environmentally relevant concentrations because they can still have negative consequences. The theme of this talk, dilution, doesn't necessarily fix all of these challenges. But beyond this, I think it's important to acknowledge that these organisms are embedded in ecosystems that may vary in different other things like water chemistry. How do these different environments with different water chemistry influence the effects of salt? Now, one way that water chemistry can differ from location to location is through the input of leaf litter. We have plant species that have leaves that vary in chemistry. When these leaves enter into water aquatic ecosystems and break down, they can also affect the chemistry of the water. And so in this section, we'll ask, how do shifts in water chemistry modify how toxic salt is? To do this, we actually focused on invasive species. Many aquatic ecosystems face challenges from invasive species, and invasive species have chemistry in their leaves that make them more susceptible or better able to compete against native species. When these leaves enter into the ecosystem, how does this shift in water chemistry affect an organism's susceptibility to salt? And so the approach was really quite simple. We went out and we collected leaf litter from 6 different plants, 3 invasive in red and 3 native. We soaked the leaf litter in water. We collected this water and placed them in a beaker. In each of these beakers, we had a single egg, either an egg from a leopard frog or from the African clawed frog. We let them grow up in this water until they reach the tadpole stage. And then we asked, how does growing up in these different environments influence their susceptibility to salt? So here are the data for this experiment. On the x-axis on the bottom there, this is native or invasive plant. That was the environment that they grew up in. And then on the y-axis is the tadpole susceptibility to salt. So a lower number here means they're more susceptible. A higher number means they're more tolerant. So for the leopard frog, what we found was something about that growing up in invasive leaf litter made them more susceptible to salt. But for the African clawed frog, it was the opposite pattern. Something about the invasive leaf litter made them more tolerant to salt. And we've since explained this, but for the sake of time, I will just leave you with the conclusion that the toxicity of salt depends on shifts in water chemistry. So not only do we have to think about all the other organisms that wildlife are interacting with, we also have to think about the non-living factors, such as water chemistry, that might interact with road salts to either exacerbate the consequences of salt or dampen the effects of salt. So I know that I just presented a lot of work here, so I'll try to conclude it all here. We study the effects of road salts on wildlife. And one of the conclusions I hope that you can take away from this is that we need to increase our complexity in the types of studies that we run because it matters. That variation matters in making predictions and understanding what road salts do. One model is not going to fit all. We also have to consider these low levels of road salts. These low levels can have negative impacts, and they may not be as clear as lethal effects, but they may still have harmful effects by influencing how organisms deal with other enemies like parasites. And then lastly, it's not just the living organisms. We need to think about how water chemistry and non-living factors of the ecosystem influence how toxic salt can be. So with that, thank you so much for your attention, and I hope that I can have some time to answer questions that you all may have.
Right. Thank you so much, Jess. That was incredible. So as Jess said, if there are any questions, you can type those. It looks like a lot of people are able to access the YouTube chat. So that is a great way to put the questions or comments out there as well. I am going to put my email into the chat too. So wisaltwise@gmail.com. If for any reason the chat isn't working, you can email me. Yeah, I did see Carolyn was able to get to some closed captioning. So I will be digging into, or Carolyn, if you want to email me afterwards, let me know how that worked for you so I can let others know in the future. So it was, some technical content here, but Jess did a great job of kind of making it accessible for most of us, I hope. And yeah, Mark just said this is fantastic. Synergistic effects are so difficult to study. So yeah, very much agree that I think there was a question just yesterday during the evening presentation about, like, what is the, chronic toxicity standard? And it's hard to tell people, well, there's this number, right? The EPA, 230 milligrams per liter. But that doesn't tell us, the full story of what's going on and what we need to be aware of. So thank you, Jess, for just kind of diving a little bit into the complexity of that.
Yeah, no problem. I feel like we answer one thing and 100 new questions pop up.
Yeah. So is the African clawed frog that's doing better in these invasive, I guess, environments with invasive plant species? You said that that frog is invasive itself to, I guess, to what area? To parts of
Yeah,
Wisconsin or yeah.
the African clawed frogs, there are populations, some of them are in Southern California. I don't think that they would be too successful here given the cold. But organisms will be able to adapt over time. And so I think thinking about it broadly, it's interesting that when we test for the effects of salt, we tend to test it in perfectly clean water. And so I think that the major point that people should consider is that, like, what does that actually tell us when we're thinking about the effects of road salt in environments that very much differ in chemistry from one location to the next?
And I try to like use the analogy for people. It's like, yeah, you can do something, right, that stresses you out, right? Maybe you're drinking water that's too salty or, what have you. But it's one thing if you just get to sit in a recliner all day long, right, and kind of be fed food and have the water brought to you. But it's another thing if you're drinking that salty water and then you also have to go, run 5 miles and, chase your 2-year-old and then, get food on the table and, that your stress level from all those other things goes up, right? So we do have some questions here that have come in. So Alice has asked, with so many variables to consider, is it impossible or incorrect to make general recommendations for limiting salt?
I think that is a question that we as scientists grapple with every day, right? Like we have to balance what information do we know and how do we use our best understanding of what that does, and that's what those LC50 tests do. I don't want to give the impression that these aren't valuable. I just want to communicate that we need to understand what the limits are so that we can fully understand what these salts are doing. So I think what we are using now with those toxicity assays are a great benchmark. But understanding that we should probably integrate in more of that complexity.
Thank you. Another question, is this a common trend of invasive species allowing other invasive species to do better in native regions?
Yeah, this is a theory that a lot of people who are interested in invasion biology think about. It's called the invasional meltdown hypothesis. It's the idea that invasive species will facilitate other invasive species. And there's mixed evidence for it. There's evidence like this one where the invasive leaf litter made it easier for the African clawed frog to persist when faced with salt. But one thing to consider is that in this study, maybe we only used 3 invasive plants. Maybe there's just something about those 3 invasive plants that changed water chemistry in a way that was beneficial to the African clawed frog. Like it's not necessarily, I think, in this study, invasive or non-invasive. We really need to dive into like the chemistry. Maybe there are some native species that have the same chemistry. We just didn't test it.
All right, another question. How does the amount of salt in your examples compare to the 230 milligram per liter chronic toxicity limit?
Yeah, so we were up much higher. So the highest concentration that we used here was 3 in the sublethal. So that would be 2.25. So they were about 4 times higher. The lowest concentration was 4 times higher than that chronic value that you're referring to.
Great. Adele has asked, or maybe I should read the question first, maybe a comment. Very curious. Okay, yes. If there's evidence that salt could be magnified through trophic levels such that predator species in a higher trophic level could be ingesting a higher concentration of salt.
Wow, I've never been asked that question, and that is something that sure happens with a lot of other pollutants. I'm not aware of research that has looked at bioaccumulation of salt, but my initial guess, based on what I understand about how organisms are dealing with salt, is that there's only a certain amount that the organisms can tolerate, and so a lot of it is excreted out. So my guess is that bioaccumulation is unlikely because it is leaving the system. That being said, there's a lot of research and understanding of how fish deal with salt. When it comes to amphibians, it's actually we just had a symposium in Malaysia that we surprisingly know very little about how many organisms deal with salt from a physiological standpoint.
All right. Well, you were thanked for an excellent presentation. Next question, did your research look at only road salts? Did you look at agricultural salts or any other type of salts?
Yeah, I should have mentioned in the research that I presented today, I focused on sodium chloride. We've also looked at magnesium chloride and calcium chloride, and um, we have looked at how water hardness can also influence susceptibility to salt. So in that invasive native study, one of the things that differ between native and invasive species is how they change water hardness. And so to follow up on that study, we, instead of using leaf litter, we just changed water hardness and we were able to find the exact same result, which suggests that why native frogs were harmed in invasive leaf litter is directly linked to water hardness. So yeah, great question. There's also a lot of variation in how toxic these different salts can be.
Another compliment here that you did an incredible job walking us through complex studies, helping us understand both methods and conclusions. Many thanks for your continued work.
I appreciate it.
Um, Another comment here, I think when reading or talking about salt toxicity, it's important to pay attention to whether quoted concentrations are as sodium chloride or magnesium chloride or just the chloride ions.
That is absolutely true.
Thank you for your valuable information. Next, we have a question. How much reduction in road salt use would need to occur to reduce stress on ecosystems? Is it practical for communities to reach that with current road management options?
Ooh, that is a great question. And I think out of my immediate expertise, I can draw on my conversations with the folks at Madison Streets Division. I think it's difficult because not only do they have to think about how much salt goes onto the road, it's how it's getting into the different aquatic ecosystems. And so it's hard because it depends on the topology, the climate. And so I am going to punt and basically say come to the talk for the rest of the week because I will certainly also be learning a lot about that particular question.
Yeah, and I mean, I think the work of Wisconsin Saltwise and a lot of the other partners behind this week see that we do have a number of options right now to drop salt use without really impacting level of service that public agencies provide. So I think in the email that I sent out with today's link, the second article highlighted Oak Creek, which is a municipality in the Milwaukee area, and Tyler Berger there, who runs their DPW, said that they've dropped their salt use 50%. I know in the Madison area, I think it's about 40% total reduction in salt use, like annual salt use in the last maybe like 5 to 10 years compared to what they had been putting down. And I mean, you can look at this, you have to try to normalize it according to winter weather severity, right? But 40%, 50%, right? I mean, these are huge reductions, substantial. And I think a lot of municipalities out there that haven't really started to dial in salt use have a lot of low-hanging fruit. But then there's also, it's not just municipal salt use. We have to look at salt going down on the private side as well. So a lot of education and outreach around utilizing those best practices that we'll hear a lot about tomorrow. And then, okay, someone knows you. Jasmine says, hey, Jess, have to ask a question about the parasites. To add more complexity to the system, do we know the direct effects of salt on parasite survival? And thanks for the great presentation.
Yeah, that is a great question. We have in the lab focused on both the effects of salt on the host as well as the effects of salt on the parasite. From what we can tell, there is a limit to where the salt is going to cause mortality on the parasite. At the environmentally relevant concentrations, we're finding that these parasites are still able to get in. And what we don't know yet is when they get in, are they still viable? So that's sort of where we're heading next. We also know that for the parasites in particular, they're very susceptible to differences in the type of salt. So sodium chloride is very different than magnesium chloride, very different from calcium chloride. Altogether, though, between the host and the parasite is going to be more susceptible to salt than the host.
All right. And Josiah, who is one of your students this fall, says it was so wonderful to listen more in depth about this. So, um, all right. And then we have a question. What type of wetlands are seeing sodium chloride concentrations of several grams per liter? I think you said that those are biolot or environmentally relevant. So kind of where do we see that?
Yeah, one of the locations in the Madison area that get a ton of salt, like it's in a depression close to the hospital, and something about that particular location has incredibly high concentrations of salt. One of the things that I'm not sure is where that source is coming from, and that's something that UW-Madison is really fortunate in that we have what we the Northern Temperate Lakes long-term ecological research. And one of the things that we're really hoping to understand is how chloride concentrations are changing over time and across the landscape. What are different factors, urbanization, road densities, how do these different things contribute to the concentrations of salt? So there are locations and ponds where we get really, really high concentrations. But at this point, I couldn't tell you what is driving that increased salt concentration. But maybe,
Sounds like if it's kind of right by the UW hospital, right? I mean, there's a lot of pavement, right? And a lot of salt going down in those parking lots.
yeah.
Yeah. All right. Well, those are the questions I see right now in the chat. I'm just going to double check that we don't have any that came in. Okay, we do here from Gmail. So Leslie is asking, are there any observations about predators that are the same, sorry, that are in the same environment and consuming their meals from animals or plant life that are in the same environment and at different levels of sustainability of salt? Also going up the food chain, consuming organisms that have consumed others and so on.
Yeah, I think that's a good question and very similar to a question earlier about bioaccumulation. I don't know of any evidence of bioaccumulation of salt. I think the reason for that is organisms are excreting the salt, the excess salt. So it might be in the ecosystem, and there's a lot of research thinking about the legacy of salt pollution. And so as it changes across time, it flushes and salt can come back up. But I am not aware of research in bioaccumulation over multiple trophic or different from it, like the predator to the prey that it eats. And again, that's probably because of how I'm understanding and what we know about the organism excreting the salt.
Looks like there's a question from Ethan about salt truck cleaning, but I think I'm gonna let Jess off the hook for that one. And Ethan, I'll send you an email kind of response. And then I'd say also, hopefully you can join in on some of the presentations tomorrow with some of the Department of Public Works staff. So yeah, with that then. I think we're good, Jess. Thank you again so much. Yeah, I want to just thank everybody who joined in today and stuck on. I know a little later here with the questions. I am hope to see a lot of you on tomorrow. We have tomorrow very interesting, like a whole bunch of webinars actually throughout the day with folks from a variety of different states. A couple of those webinars do require you to register. I think that's the Ohio and the New Jersey webinars. But others I think are just available like today's with just a quick link to YouTube. So thanks for joining and thanks again, Jess. And yeah, happy Winter Salt Week, everybody.